There are many cases where it's not practical or possible to label the experimental sample. For example, when working with tissue samples, or performing large fermentation runs, or propagating field-grown plants. In these instances, the Iroa Phenotypic experiment may be applied. The cluster finder software algorithms have also been written to find the control Iroa patterns in molecules and then search for the unlabeled experimental metabolite pairs. Therefore, two Iroa protocols are possible. The basic Iroa protocol, which is an untargeted method whereby control samples are labeled with 95% C13 media, and experimental samples are labeled with 5% C13 media, and a second protocol, called the phenotypic protocol, where the control samples are labeled with 95% media and compared to their natural abundance counterparts. For both protocols, the Iroa cluster finder performs a full analysis, removes all artifactual peaks, and identifies and quantitates the remaining metabolic peaks. In the phenotypic Iroa experiment, the control or standard is isotopically labeled using Iroa C13 media. An ideal standard is one that represents the entire metabolome of the fluid or tissue under study. For example, if interrogating the metabolome in breast tissue, quantification of metabolites can be achieved using an Iroa-grown breast cell line as a complex internal standard. The complex internal standard is spiked into each experimental sample and processed as a composite so that the technical variability is removed. During the phenotypic analysis, the cluster finder software will use the C13 Iroa peaks in the labeled control sample to identify their associated natural abundance peaks in the experimental sample, allowing for a complex targeted analysis. Using the basic Iroa protocol, where both the control and experimental samples are labeled, the Iroa signal is made up of two halves, the C12 half and the C13 half, in which the carbon envelopes are mirror images of one another. With the phenotypic Iroa protocol, the experimental sample is unlabeled and contains only natural abundance carbon. Here, the cluster finder software identifies the C13 label peaks and then calculates where to find the corresponding C12 peaks. The pairing makes the identity of all the peaks measured unambiguous. Using an Iroa labeled standard, all of the peaks may be easily identified according to the presence of their characteristic M-1 peaks. The natural abundance peaks corresponding to each standard peak may be readily identified because even though they do not carry an isotopic labeling, their exact mass and position are established relative to the standard. The distance between the paired base peaks defines the number of carbons in each molecule. If the pooled standard is well characterized and the compounds that are present in it have already been identified, then it will be able to be used as a point of comparison for all of its contained compounds. Artifacts will have no match in the standard and are eliminated by cluster finder in the final data set. In the phenotypic experiment, all experimental samples are measured relative to the standard. Therefore, the phenotype is defined by the deviation from the standard.